To understand how nucleotides are arranged in a DNA molecule and even in an RNA molecule, first we need to know how carbons are numbered in the pentose sugar. So the 5 carbons of a pentose sugar are numbered 1 to 5. The carbon that is attached to the phosphate group is carbon number 5. And then you go clockwise, so the one on its right is number 1, and then 2, and then reaching 3. And carbon number 3 has a hydroxyl group. This is how we number the carbons of a pentose sugar in a nucleotide. If we're talking about DNA, we have 2 strands of polynucleotides. These strands are anti-parallel, which means they are parallel to each other, but they run in opposite directions. One of them is going to run in a direction we call 5' to 3' direction, while the other runs in the opposite direction, so the 3' to the 5' direction, because they are anti-parallel. The one running from 5' to 3' starts with a free 5' end and ends with a free 3' end. And the one on the other side is going to start with a free 3' end and end with a free 5' end. In the body, nucleotides are synthesized from the 5' to the 3' direction, which means nucleotides are going to be added to each other at the 3' end. This is why the last nucleotide has a free 3' end. They are synthesized from a 5' to 3' direction. And this is simply because the enzyme that synthesizes DNA, which is DNA polymerase, can only assemble nucleotides this way. It attaches nucleotides to hydroxyl groups, and we just said that hydroxyl groups are on the 3' carbon of the pentose sugar. The 5' is attached to a phosphate. So nucleotides are synthesized from the 5' to the 3' direction. And in DNA, the 2 strands are found anti-parallel to each other.